it is just the when the initiator tRNA is binding to the initiating codon and with the initiation factors and the ribosome, that acts as a stimulus. So there is no distinct gap there.
Yeah.
Okay, sir. Okay, sir. Clear.
Good morning, sir.
Morning, Kuna.
Sir, I have doubt. I think like is it related or not, sir? When after the charging of tRNA, sir, when I detect the codons, sir, every charging tRNA is crowded surrounding the ribosomes. Because every time they are checking that is the correct anticodons, then it is not, then it will away from another charging tRNA will check.
So I am not sure whether I clearly understood your concern, but tRNA charging, the fidelity of that or gatekeeping of that is at the level of amino acid tRNA synthetase.
Yes, sir.
So there, because the amino acid binds first, and then only it creates the appropriate opening for the tRNA. So yes, there will be multiple tRNAs competing for the site, but only the most appropriate one will be able to get charged. And during elongation, yes, multiple charged tRNAs will attempt to enter the A site, but only the one with the appropriate anticodon loop will actually be able to enter. Like not enter, get stabilized at the site. But you should understand that the rate at which protein synthesis happens is like, I think 5 amino acids per second or something like that. It is quite processive.
Yes, sir. That's the. Yeah. That's I have doubted. It is time taking, like.
No, yes. It is not time taking. Like time taking in certain scale, but if you think about it, that everything is happening in the molecular scale, right?
Yes, sir.
So a ribosome whose size will be perhaps few nanometers, and it is moving on the mRNA few microns per second. Now you extrapolate it to real life moving objects, you will see that the speed is actually quite high.
Okay. Yes. Okay, sir.
Sir, can you explain once the Nienberg and Leder experiment? The one.
Which one? The the filter one?
Yes, sir. I have that doubt with the filter one too.
So it is fairly straightforward that charged tRNA. Let me open the slide.
Yes.
The pore size in the filter paper is large enough so that charged tRNA, like what is shown here, I forgot to bring my pen today. The charged tRNA here can actually pass through the filter. Okay. Ribosomes cannot pass through the filter, but they are not charged.
Okay.
Now, the only way this tRNA will be retained on the filter paper if it is in a complex with ribosomes.
Yes.
And that can happen only if the ribosome is primed with corresponding artificial codon.
Hmm.
So that's how.
So
So
this experiment showed that the specific codon is actually bind by the specific anticodon loop from the tRNA, and this is complex is with the ribosome.
yeah,
Okay, okay, sir.
Sir, all this was done in test tube. Yes, sir.
yeah, this was done just to demonstrate the entire range of 61 codons.
Yes,
Hmm.
sir. Sir, the earlier experiment that they have did, sir, they have used this sequence.
Mhm.
So here, did they need to start the translation? They need start codon or ribosome binding site to how
Very,
directly
yeah,
started
very,
with this?
very good question. Excellent question. You were the first one to ask me this question in 14 years. Um, it is actually the scientists in those days, the biochemists, they were extraordinarily ingenious. So you are correct that this artificial mRNAs, they did not have either start codon or Shine-Dalgarno sequence. How did the ribosome even start crawling over it?
Yes, sir.
The answer to that, and with some bit of pride, I can tell you that Hargobind Khurana spent 3 weeks in my PhD mentor's lab to perfect this part. Okay, so the solution is an extraordinarily high concentration of magnesium. Okay,
Okay, sir.
so that's how they artificially made this recruitment to happen.
Sir, so
So,
like when the concentration of Mg plus 2 plus is very very high, then the association of larger subunit and smaller subunit will take place.
Not only larger subunit and smaller subunit, in the context of mRNA and tRNA and everything.
Okay, okay, okay.
See, you cannot explain this by the same yardstick of normal translation initiation, right? Because normal
Yeah.
translation initiation, you will have first the 30s and the 30s initiation complex, scanning, AUG recognition, then 50s, then polymerization. Uh,
Hmm.
I don't think anybody has studied how exactly this happens in this artificial system.
Yeah.
okay, sir. Sir, so in both these experiments, they also didn't need their initiation factors, elongation factors.
So all those things were there in the cytosolic extract, right? The
Okay,
cytosolic
okay.
extract had everything other than RNA.
Okay, sir.
So in their language, they were programming the cytosolic extract with one artificial RNA or the other. I could not find the reference, but I want to believe that they perhaps treated it with RNAs to degrade the existing like endogenous RNA, or
Okay.
or some other trick. Like they must have depleted the pool of endogenous RNA somehow.
RNAs. Yes.
Sir, but they are depleting only the mRNA, not the tRNA or the rRNA. In
Yes.
the extract, है ना? They
Ah,
are yeah. So there should be a mRNA specific degradation something method they have.
yeah. So mRNA degradation का तो कोई method है नहीं.
Yeah.
So I do not know. I also tried to look for it that how they are depleting the mRNA pool. See, in eukaryotes, it is relatively easier to think using a like a oligo-DT column.
Hmm.
But in
Yeah.
prokaryotes, I really don't know how they managed to, because see, if the endogenous mRNAs were there, their experiments would not have worked.
Worked. Yes.
Okay,
Yes,
so
sir.
don't know.
So like these experiments were very interesting. Like we till now we were never taught these experiments that actually how it went on
Huh.
happening. Yeah.
Yeah, I know
The
that.
like that part of Hargobind Singh Khurana was like touched upon, but this part of Nienbag Lede was never touched upon. So it was very
Mhm.
interesting.
Thank you.
Yes, sir.
I
Sir,
used to
you
teach a course of 5, 6 lectures of history of molecular biology.
Yes.
I no longer do that.
okay. Sir, you also talked about nucleotide composition. Sir, how is it different from nucleotide sequence?
See,
Yeah,
like
yeah, sir.
we are talking about an era when a nucleotide sequencing method was still not discovered, right?
Yeah.
Yes,
So
sir.
all they would do is just depolymerize it and run some kind of a paper chromatography to figure out the percentage of A's and T's and G's and C's.
Okay, okay, okay. That's how.
Yeah.
Yes.
It is very similar to what this guy did with DNA. मेरा problem क्या है ना किसी का नाम नहीं याद रहता है. अरे this like who first Shargaf, right? A is equal to T, G
Yes,
is
sir.
equal to
Yes,
C.
yes, sir.
Yes,
Chargaff
sir.
Road.
Shergraph.
Yes, sir.
Yeah, it is very similar to that.
Okay, sir.
ये मेरा बहुत बड़ा problem है. I just can't remember any name. You ask me facts, I can remember. Names I can't.
And sir, here also the filter paper was used to separate the polymerized stretch from the non-polymerized stretch. No?
Yeah,
Okay.
yeah. Like
Oh.
I have done a whole lot of these acids. So
Hmm.
you can do it in one of two ways.
Hmm.
Either by filter detention
Yeah.
or you can use trichloroacetic acid. Trichloroacetic acid will precipitate all the proteins. Okay.
Yeah.
And then you wash it with acetone a lot, and then everything will be washed off other than proteins that are precipitated. And you can count the radioactivity in the precipitate.
Okay.
Like the, like there used to be this machine. Nowadays, I don't think any lab has those machines anymore. It's kind of a fluorimeter, very large machine, very large. Like, have you ever seen those um, uh, refrigerators where they keep ice creams, like the horizontal refrigerators?
Yes, sir.
Yes, sir.
It used to be of that size, and there would be a conveyor belt. Okay,
Hmm.
and then you add a liquid. I'm now forgetting the name of the liquid. It has been a while now. Uh, that liquid was nothing but this that radioactivity, uh, like it will emit some radiation, right? That will hit one of the components of the liquid, which will emit light, and that would be detected. Okay, so that's how these experiments used to be done.
Sir,
Mm-hmm.
sir, in this experiment, how did they identify that the poly-U stretch is composed of phenylalanine, polyphenylalanin, and the poly-A stretch is composed of polylysine? How
So,
did
so,
they
so,
differ?
so
How
very
did they make
simple,
difference?
very simple, right? You take 20 tubes
Okay,
and you prime it with poly-U in everyone.
okay,
Okay.
okay,
And then in each tube, you add one or the other amino acid radioactive.
okay.
So you will have 20 different amino acids radioactive.
Yes,
Only in one tube you will get polymerization,
okay,
where you have phenylalanine radioactive or lysine radioactive. That's how it was done.
okay, sir. So by the radioactivity level, just they confirm this.
Yeah. Radioactivity in the polymer.
So because I am concerning about the poly C stretch which gives rise to polyproline, how
Uh,
did the polyproline the protein folds because the composed of different proline sequences.
yeah.
So
So they certainly were not worried about folding, etc. They were merely looking at polymerization. These experiments worked. It is a wonder. But thank God they worked. That's how we got to know so many stuff.
Sir, you said that there are 20 specific amino acyl tRNA synthetase. Sir, like, are there 20 specific enzymes, or it is like the same enzyme with the association of the particular amino acid, it becomes specific?
No, 20 distinct enzymes.
Okay, 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 sir.
And it is a classic example of induced fit model.
Hmm.
Sir,
Yeah.
with respect to this amino acyl
Hey,
tRNA,
Shreya, did I interfere with your Bhaipota plan?
no, sir, no, sir.
Thank God. I was worried some people will kill me.
No, sir. So my question is with respect to this amino acyl tRNA transferase activity. So, you said that there is a specific enzyme for a specific amino acid.
Yeah.
So, like, there are some amino acids where there is very like fine change. Like, if we consider valine and isoleucine, so
Uh,
there's only a change with one.
even then, this is that stringent.
Okay, so such stringency is observed even in the prokaryotes, or
Yes, yes. But you know one very interesting stuff, slightly unrelated, but you may love to know this. So, like as you know that my PhD training was in protein synthesis, right? So my job was to look at formation of those pre-initiation complex through filter binding assay and initiation complex through sucrose gradient. Okay, so I needed to have uh, methyl tRNA charged with methionine, radioactive methionine. Okay, and that is the only experiment where my PhD mentor used to do it himself. Okay, and you don't want to know how the lab used to be for those 2 3 days, uh, but like these old-fashioned biochemists, they were so knowledgeable you cannot imagine. So they knew, and it is such a wonder, they knew that if you take a bacterial extract, cytosolic, like bacterial extract, तो cytosol nucleus अलग नहीं होता है. Bacterial extract, the only eukaryotic tRNA it can charge is that methionine tRNA, initiator methionine tRNA. So the experiment was simple. They would get a crude tRNA mix from rabbit reticulocyte lysate. They will take a crude cytosolic extract and radioactive methionine. But because of the specific, crosstalk, only the methionine tRNA synthetase of the bacterial extract could charge the eukaryotic, like rabbit reticulocyte tRNA with methionine. So what we used to call as charged methionine tRNA, if you look at my papers, I have used it a lot. Those are actually not radioactive, not pure methionine tRNA charged. Those are, contamination of all sorts of things, bacterial extract, eukaryotic extract. But the only thing that was charged was this tRNA. So yeah, like the specificity is really stringent and interesting.
so, sir, can it happen like by chance a wrong amino acid gets charged? So then, in that case, will it be removed, or that entire
No,
tRNA complex it
no,
will
it
be
will be
maintained?
incorporated. So you can imagine a very simple experiment that you can think about. You take tRNA charged with cysteine. Okay, cysteine has a sulfhydryl group, right?
Yeah.
So if you oxidize it further, it can become serine, right? So if you do that, then in place of cysteine, the protein will incorporate serine.
So, sir, like there is no further proofreading mechanism.
No, There is no further proofreading mechanism. That's why the amino acid tRNA synthesis is so critical.
Okay.
Yeah. Like uh, we were extremely fortunate while my PhD mentor was studying protein synthesis. He was, if you look at his old papers, he's the guy who discovered T3 and T7 RNA polymerases. Okay, as a postdoc with his postdoctoral mentor, and the lab next door used to be of Dr. Ladan Schulman. She was the authority on tRNAs in those days. Like a whole lot of work that today we talk about, these identity elements, etc., came from Ladan's lab. Okay, so yeah, we were extremely fortunate to learn from the masters.
So one more thing, like you said, since like that time sequencing was not that developed, like the technique of sequencing. So how did they actually determine these identity elements in the tRNA molecules?
Slowly, those things came later.
Okay.
They did not happen at the same time. They happened much later over a period of time. Can someone remind me when did this Sanger sequencing start? It must be early 70s, not before that. Let me quickly check. In 1975. It was 1975. So you can imagine that when all this work was going on, sequencing of DNA, RNA was far from its scope.
Mhm. So that day I had asked a question like once these mRNA are transported, so they
Okay,
are.
so I could not manage to load the other thing, but let me give you the answer. A little bit, let's go into a little bit of philosophical discussion. Can someone open the laptop uh, and find the average size of a protein in angstrom? Oh, I forgot. Okay. No, I did not bring my pen. Okay, forget it. I'll tell you without the pen. Please visualize along with me that Ubaid was also asking, or Kuna was also asking. All these questions relate to the same thing. The Antara,
Yes, sir.
ভাই ফোটা হয়ে গেছে?
না ভাই, ভাই এখানে নেই।
Okay, so over internet. In my home, lots of futa happened in the morning over internet. So, what is the average size of a mammalian cell?
Sir, 1 minute. Sir, you're not clear. 1 minute.
What is the average size?
Sir, can you please repeat?
Yeah. What is the average size, that is diameter of a mammalian cell?
Um, Sir, like 100 micrometer.
No,
Yeah.
10 micrometer. Okay, that is the
Oh,
average size. Okay,
okay.
so and who can tell me what is the average size of a protein? You know, a relatively large protein.
So like hemoglobin molecule is 5 nanometer.
Okay, so now we have two numbers. Okay, uh, hemoglobin is 5 nanometer and a human cell is 10 micron. Okay, now let us think in terms of volume. So 10 micron, like the volume will be pi r cube, right? Without getting into, you know, like dividing 5 by 2, वो भी आप कर सकते हो. You can imagine that it is 5,000 cube. That means 9 zeros after 5 divided by 2.5 cube. Okay, like let us say 5, like 125. Can you imagine the order comparison?
Right.
So it is perhaps will be much more than the ratio of a football to a football field.
Mm-hmm.
Now visualize what is the probability that two molecules will collide by chance?
Very low.
Very, very low, right?
Mm.
So, like if I had recorded lecture from 2007, I used to talk about it in my class in those days as well, that I firmly believe that there are non-membranous compartment in the cell that ensures that the molecules that are supposed to work together are localized within a particular region. There are micro domains within the cell wherein molecules belonging to the same pathway or related, they will be together. Like anyone here planned joining Nitin Mohan's lab? No one? Okay. So I have been discussing with Nitin that a proof of this principle would be that in eukaryotic cells, if he does super resolution microscopy analysis of relative localization of, say, enzymes of the glycolytic pathway, I'm pretty sure he will find them clustered. Now, I will tell you about a very old data, very old data. So some of you may have this notion that the mRNAs come out of the nucleus and they are kind of floating around and ribosomes latch onto them and translation happens. If you use radioactive amino acids, you just grow an eukaryotic cell and ensure that all the amino acids there are radioactive in the growth medium. And then after growing it for 5, 10, 15 minutes, you stop it and lyse the cell gently. You gently lyse the cell and centrifuge it. Then you will get the cytoskeletal components in the precipitate and everything else will be in the supernatant. Could you hear me, Shreya?
Yes,
My,
sir.
huh?
Yes, yes, sir.
Okay.
Yes,
Then,
I could hear you.
then when radioactivity was analyzed, it was observed that 85% of the radioactivity was associated with the cytoskeletal fraction. What does it mean? It means that you are adding radioactive amino acids as a pulse change, right? You are just pulsing it. So that means that the freshly initiated polypeptide chains are being produced. You know, I showed you those polysomes.
Yeah.
So those polysomes are firmly attached to the cytoskeletal network. Point number one. Point number two, when you think of neuronal cells, okay, the cell body is sometimes meters away from the end of the cell where the synapses happen, right? Now, for the synapses, you know, for proper communication from cell to cell, mRNA, the proteins have to be present on the cell membrane. Now, the way it works is that the mRNAs, after synthesis, they literally ride on a train called dynein motors and travel all the way to the end of that like synaptic end and are stored there, and on command those RNAs are translated and proteins are formed and they become part of the cell membrane or right below it. Okay, then I will show you data that when yeast cells divide by budding, there is a particular mRNA, a protein that is required only at the bud site. And there is real time video imaging that how in the beginning of budding, the mRNA is actually carried to the bud site on the back of motor proteins. So if you take everything that I said so far together, in my mind at least, the picture that emerges, and by the way, in mRNAs, certain signature sequence has now been discovered called zip code sequence. You know, in the US, the postal address, like we have pin code, they have zip code. These zip code sequences are apparently responsible for deciding the subcellular localization of the mRNA, not the protein, of the mRNA. So, to me, it appears that the RNA comes out of the nucleus, immediately gets a ride on one kind of protein or the other, and there must be some linkage between the proteins that are wrapping the RNA and the proteins that will load it onto this cargo, onto those motors, and that will dictate which part of the cell the mRNA will get trafficked to. And I further speculate that the mRNAs belonging to the same pathway, they will be trafficked in the same to the same destination. Okay, so were my hypothesis for a long time, but now if you study the literature for last couple of years, you will see a whole lot of articles coming out which refer to something called phase separation. Okay, so there is some sort of phase separation in the cell. Okay. I have not been able to read those papers carefully, but I think they are talking about the same things. Okay. To that, you add another dimension which Gunther Blobel talked about, gene gating, that the three-dimensional architecture of the DNA chromatin. He speculated, like when I will teach protein trafficking this week, I will load the lectures. If there is, of course, there are way too many brilliant scientists, that we all know about. But if I have to pick my one hero, that would be Gunther Blobel. This guy had so much conviction in himself. He proposed this entire signal hypothesis when he did not have an iota of evidence for anything that he is proposing. He proposed purely based on logic. He said that if this is how it is happening, this is how it has to happen. Okay. And he proved it 25 years down the line. Similarly, in those days when imaging techniques were not at all that advanced, he speculated that the three-dimensional architecture of the chromatin will change from cell type to cell type. Like changing the three-dimensional architecture will be a part of differentiation process. Now, connect that to what we just talked about. I will not be surprised if the status of the cell, the differentiation status or whatever, status of the cell that dictates the 3D architecture, that dictates the location within the nucleus where a certain mRNA will be made. Perhaps there is segregation already in terms of which mRNA will come out of its site. So, because see, like the speed at which things happen, chance collision is almost out of question. Almost out of question. Like I can show you data that, by now I think you all know that I work with BMP signaling pathway. So if I add a BMP ligand and there is a reporter that reports BMP signaling activity, I can detect the reporter gene expression, which is an enzyme that emits acts on this substrate called luciferin and it emits light within half an hour. Now imagine what is happening within half an hour, that you add the BMP ligand that binds to the receptor gets phosphorylated, that will phosphorylate a transcription factor resident in the cytoplasm, that will then move to the nucleus. It will transcribe a whole lot of genes, including the reporter gene that I have given. That will be transcribed, mRNA will come out, it will be translated, the protein will form and become active. All of this is accomplished within half an hour. Okay. So, there are, I really think that the idea that within cytoplasm things happen by diffusion and random collision, I think that idea is now being severely challenged. I think it is stratified from the word go. Okay.
So this subcompart. Yes, sir.
Yeah.
So this subcompartmentalization, like when they observe the polyribosomes and all
Yeah.
through electron microscopy,
Yeah.
so that time they did not get any hint like from the electron microscope images, or like such
See, those days they did
resolution
not
is
do
not
electron
attained.
microscopy. No, in those days they did not do electron microscopy. They simply detected radioactivity either in the solution phase or in the precipitate. But yes, nowadays that's where people like Nitin Mohan comes in play. So their super resolution microscopy techniques. See, the problem is that um, someone needs to know that these are unanswered questions in biology, because the technology development happens in one direction, and classical knowledge remains in another direction. So these two worlds seldom meet. Okay, that's actually a major problem. Yes, Priyanshu.
Sir, what are the exact location of the protein synthesis in a eukaryotic cell? I mean, previously we have learned that the protein synthesis takes place on a rough endoplasmic reticulum where the ribosomes are located, and then there are certain free ribosomes in the cytoplasm as well. So,
Yeah.
which proteins are synthesized there, and which are the synthesized in the cytoplasm?
Yeah, so I will teach all of these in my trafficking lectures. But in a nutshell, it is believed that proteins that are destined to be secreted, they are synthesized on the rough endoplasmic reticulum, while the soluble proteins are synthesized not on the RER, but I am confident that they are synthesized while remaining attached to the cytoskeleton.
And also, sir, like there are multiple sites of protein synthesis, or there is a some fixed place where this protein synthesis takes
No,
place?
multiple sites. I would think it is multiple sites.
Okay, sir.
Yeah.
Thank you.
Yeah, Navneet? Navneet, was it by mistake? Okay.
Sir, can you hear me?
Yeah.
Yeah. So I have a question on RNA interference. I can't understand how the sense transcripts would have produced interference. Like, what mechanism does it follow?
Yeah. So, not that I understand it either. We talked about this in the last class. See, if you are making the sense transcript in vitro, there they speculate that there might be some cross contamination. But if you actually visit some of the internet sites, that has been actually challenged. But unfortunately, once theory gets prominence, the challenges do not appear prominently. Andrew Fire and Craig Mello speculated, but they never demonstrated that there is contamination in the in vitro synthesized sense strand, or they offered no explanation that how in vivo, which Andrew Fire himself did, uh, synthesis of the sense strand can possibly generate some double-stranded species. So
Okay.
yeah, it was
So we still don't understand how it does that.
yeah. So neither do I. I do not think anybody understands it.
Okay.
But sir, doesn't it mean that I mean it is very hypothetical, but doesn't it mean that there might be some transcription in the 3' to 5' direction as well, if sense strand is imposing some interference?
Well,
I mean, one of the things can be that as well, but that is
yes,
very hypothetical for the
yeah,
time being.
Yeah, so that is a possibility. That's what people do artificially in C. elegans. If you remember in my lecture, I talked about it. In C. elegans, what they do is whatever gene they want to knock out, or now that I have taught you about how cDNA libraries are made, now just imagine that you are making your cDNA library in a p-blue script vector where on either side of the uh, multiple cloning site, you will have T7 RNA polymerase, like T7 promoter. Okay, so now if you are, like putting it inside of any bacterial strain that produces T7 RNA polymerase, so you will actually transcribe it from both the ends. And that's how they used to do screens for genes like loss of function screens in C. elegans. But that was deliberately done. What is not clear is in Andrew Fire's own experiment, where he bombarded and knocked in a certain transgene in C. elegans, how the from which is the source of the contaminant antisense transcript that will allow for formation of the double strand. One thing is clear, that even if you make one molecule of double strand, then it can amplify. That much is very clear. So the only possibility is, as you rightly pointed out, that if there is very low level of leaky transcription, because something is being recognized as a promoter like, and it does not have to be in the immediate vicinity, even if at some distance, then also that might work. So that is a possibility. I cannot rule it out. So you are correct.
And sir, also in the paper of Fire and Mellow, they mentioned that due to the double-stranded DNA, the efficiency of double-stranded DNA as an RNA interfering species might be due to the stability that it might provide uh, over the sense or antisense strand singly.
Yeah.
So my question is, on the other hand, there is a mechanism where some of the RNA transcripts are made double stranded and then they are degraded via a pathway. So how are they speculating that double stranded RNA
I
species?
think the way, like now these lectures are delivered in a very different manner. Um, I could not at all talk about the biochemistry uh, that led to identification of the whole pathway. Please remember that Fire and Mello, they only described the phenomenon of RNAi. They did not discover the components that make it happen. So all that work was done by Phil Sharp and another person in Cold Spring Harbor. I'm forgetting his name. So they later showed that dicer can only be programmed with a double-stranded RNA and not with a single-stranded RNA. So it is a substrate specificity issue primarily.
Okay, so
Anything else? थोड़ा और पढ़ो. You guys are running out of question. Actually makes me a little worried that you are not paying enough attention. Uh, there are lots of issues here to very clearly understand. Like say, if I ask you, Umar, that how would you know whether 40S initiation complex has formed in a reaction?
Sir, you mean a pre-initiation complex, right?
No, initiation complex.
Sorry, initiation. Uh, Sir, maybe some biochemical assays may be done to characterize its formation.
Correct. What will be the biochemical assay? What property would you use?
So,
How would you? Okay, different question. Promit.
Yes, sir.
I am telling you that there is a transcription factor X. Okay, X is not necessary for the translation of, sorry, I meant there is a translation factor X. Initiation, elongation, don't care. X is not necessary for translation of every single mRNA, but it is necessary for translation of a particular set of mRNAs. How would you demonstrate that? How would you investigate which mRNA's translation is dependent on X?
Then if we have identified the factor, then maybe we can inhibit it and see which genes are not being translated, which proteins are not found in the cytoplasm.
How would you do that?
Uh, we could take the whole cytoplasm extract, and if so, since we are seeing the presence of the protein, we can let's consider we have the antibodies against the protein, and then we can do a Western blot.
Against which protein?
Like, do we know the proteins that it, like, we know the set of mRNAs, right, that it translates?
No, of course not.
Okay.
So the protein that you want to investigate, which set of mRNAs they translate, I mean, which set of mRNAs it is involved in the translation. So we can develop some antibodies or maybe some that may bind against that protein. And
which protein? Which protein? Like, I absolutely detest preposition in science.
Okay, even the protein is not even characterized.
Of course not. No, X is known.
Okay.
X is known, characterized, you have antibody and anything you want against it.
We could look at the gene expression data after inhibition of X and before inhibition, like in presence of X and in absence of X.
Gene expression data, in how would you do that?
Okay, we have the mRNA, right? I got confused.
Socho bhaiya, socho. Like, do not pass the course. Okay, learn. Okay?
Sir, I was wondering, can we use like a protein microarray? Is there like a protein version for a microarray? Can we use that?
Well, slowly we are getting there, but too expensive. Let's not get there. There are easier ways of doing it.
Okay.
And I want you to figure it out yourself. Think very hard.
Okay, sir.
All sorts of ways and methods you think for both these questions that I asked Umar, that I asked Promit. Invest some more time. Okay. दोबारा कोई पढ़ाएगा नहीं ये सब subject.
So for that, to detect that whether the initiation complex is formed or not, Can we do like cesium chloride centrifugation to determine whether
Yeah,
it's formed or not?
yeah, that's how it is done. It is not cesium chloride. It is actually sucrose gradient
Sucrose.
centrifugation. And like for the 40S initiation complex, 80S initiation complex, their relative location in a sucrose gradient is known. Okay. Uh,
Okay.
so uh, I will talk about it in
So
another.
we take that as reference and then we determine whether it's formed or not.
Yes, you do. That's how you do it.
So
Okay.
for the second question, can we do two-dimensional cell analysis for both the control sample and the in which we have inhibited X, and then we can identify these
You
particular
you theoretically speaking you can, but if it is say in a standard cell apparently 5,000 proteins are made, and if you imagine 200 proteins are dependent on that X, then life becomes difficult.
Okay, so.
Yes, Ayush.
Sir, can we do like before the inhibition, let us radioactively label the, let's say, provide S35 to the cell media and allow the translation to happen. And on the experimental set, you just knock out the X and provide the radio labeled, I mean S35, and analyze what all proteins are not radio labeled in the second fraction, and then take those protein, I mean it is difficult to purify them specifically, but hypothetically this experiment will lead us to identify those proteins that are being translated with X.
Okay, that will not work. It will be the same thing as Priyanshu was trying to talk about, some kind of proteomic analysis. I'll give you a hint and I will end the class. Figure out what is polysome, what is the role of sucrose gradient in segregating different species of ribosome, and then try to think from there. Okay. I will discuss it again on Wednesday evening. We will discuss it again. Okay. And by that time, I will try to load my other lecture, the last lecture on translation. Chalo, aaj ke liye chutti. Listen to the lectures. Listen to the lectures more. Okay. Ta-da.